Hey students, so today we are going to be doing the topic 19.2 reproduction in lower plants. In this topic, we discuss about two type of lower plants. The first type of lower plant, which is Chlamydomonas, we are going to be learning Chlamydomonas, and the second type of lower plant we are going to be discussing is Spirogyra. But in today's video we are only going to be covering Chlamydomonas, and in the next video we'll be covering Spirogyra. So first thing, before we start the entire chapter about the entire topic of Chlamydomonas' reproduction, we need to understand what a Chlamydomonas is, and we need to understand its structure. Okay, so what is Chlamydomonas? This Chlamydomonas is a haploid unicellular algae found in freshwater plant. What do you mean by haploid? Haploid means it has only 23 sets of chromosomes. Okay, so what is Chlamydomonas? Chlamydomonas is a haploid unicellular algae found in freshwater pond. Okay, now look at this. This is the diagram of a Chlamydomonas. If you look closely at the diagram of Chlamydomonas, you can find out that it is pear-shaped. Its body is pear-shaped. Can you look at this? Do you see this? It has some sort of pear resemblance, right? Yeah, so it is pear-shaped and it has two flagella attached to its narrow end. What is this? This wavy thing. Okay, I'll use a different color so you can see properly. You see this wavy thing over here that goes like this. Yeah, this is a flagella. Okay, and this is a flagellum. Singular it's flagellum, plural it's flagella. Okay, so this two are attached to its narrow end. So the first thing we understand, let me write down in the corner here. The first thing we understood of Chlamydomonas is so far is that it is haploid. Okay, it is haploid. It's unicellular. And what did we learn? It's a haploid unicellular found in freshwater pond. Where is it found? In freshwater pond. Right? It's found in freshwater pond. Okay. Now, we also learned that it is pear-shaped. It is pear-shaped. If you look at its body, we learned that it's pear-shaped, right? And what else did we learn? We learned that it has a flagella, a set of flagella, or it has two flagellum. It has two flagellum attached at its narrow end. Attached at its narrow end. Now, this is the 2 points. Now, if you look closely into the Chlamydomonas, if you look at the corner here in one side of its cell, you can see this circle thing. Okay, this circle thing is called an eye spot. Okay, what is it called? It's called an eye spot. This circle thing is light sensitive. Okay, so we can also mention that towards one side of the cell we have a light sensitive spot. We have an eye spot which is a light sensitive spot. Okay, and then also if you look at the Chlamydomonas, we have learned the outside structure. Right, now let's go inside the Chlamydomonas. Inside the Chlamydomonas, you can see this cup-shaped organ here, right? This cup-shaped thing, right? This cup-shaped thing here is called the chloroplast. So inside Chlamydomonas, there is a cup-shaped large. Isn't it large? Yes. So inside the Chlamydomonas, there is a large cup-shaped what? Chloroplast. This is a chloroplast. So inside the Chlamydomonas, there is a large cup-shaped chloroplast. Okay, and towards the center of the chloroplast, or towards the absolute center of the cell, you can see this absolute center of the cell. What is the absolute center? Here, right? This spot. Towards the absolute center of the cell, what is present? A definite nucleus. This is the nucleus. Okay, So towards the center of the cell we have a nucleus, okay? And finally, if you look inside the chloroplast here, this is the chloroplast, right? What is this? This is a pyrenoid. A single pyrenoid is inside the chloroplast. We have a single pyrenoid inside the chloroplast. So this concludes the structure of Chlamydomonas. If you look here, if you read this, how many points we have here? We have 6 points. If you look at these 6 points and you look at here, then we have covered every single point of Chlamydomonas. A trick to learn the points of Chlamydomonas or the structure of the Chlamydomonas is ITOATC. ITOATC. Okay, it's a trick to learn. If you want to learn it that way, you can learn it that way. Or if you want to learn it in a different way, you can learn it in a different way. Now, let us discuss the reproduction in Chlamydomonas. Why are we discussing reproduction? Because the topic is reproduction in lower plant. What is the chapter's name? The chapter's name is Reproduction in Plants. Okay, so let us discuss its reproduction. Now, Chlamydomonas, it reproduces in 2 ways. Okay, it reproduces asexually and it also reproduces sexually. Okay, so Chlamydomonas reproduces in 2 ways. Now, in asexual reproduction, also it reproduces in different. So let's go over that again. Okay, so Chlamydomonas, okay, reproduction of Chlamydomonas is of 2 ways. Okay, the first way is asexual reproduction and the second way is sexual reproduction. Okay, in asexual reproduction, again there is 3 different ways. The first way it is zoospores, the second way is aplanospores, and the third way is hypnospores. Even in sexual reproduction there's 3 different ways, but let's just focus on asexual reproduction for now. We'll come back to sexual reproduction. Now, in asexual reproduction, it reproduces in 3 different ways, which was zoospores, aplanospores, or hypnospore. What does this depend on? This depends upon the availability of water. It will only reproduce in zoospore or aplanospore or hypnospore depending on the availability of water. Before we start and go dive deep, I'll just give an overview of what it is. So here in zoospore, it reproduces when there is plenty of water. Okay, just for you to understand. And in aplanospores, it reproduces only when there is a thin film of water. It means a very little water. Hypnospore, it forms hypnospore when there is no water available. That you will understand as we go into the topic. Okay, just know this overview for now. Now, let us start with zoospore. Now, if there is a lot of water available for swimming, okay, Chlamydomonas reproduces through zoospore. I told you if there's so much water available for swimming, it reproduces through zoospores. Now, one thing about zoospores we need to understand is that they are flagellate and they are thin-walled. Okay, flagellate meaning they have flagella. Here we have Chlamydomonas example. Okay, example here I have Chlamydomonas. I'll draw it as a circle right now. Okay, I'll just little like that. Ignore my drawing. Okay, there's no drawing skills here. Okay, now I'm just gonna draw this like this way. Okay, just imagine that is our Chlamydomonas. Now this Chlamydomonas, this is zoospore. Imagine this is our zoospore. Okay, so it has flagella. So this is why we say it is zoospore is a flagellate thin wall. It has a thin wall and it is flagellate spores. So this is zoospore. Okay. Now, how does zoospore form and how does Chlamydomonas reproduce through zoospore? Now, first thing is asexual reproduction. It occurs with the parent cell. So now let me take a parent cell. Okay. So suppose here I have a parent cell. Okay, this is Chlamydomonas parent cell. Okay, I'm just going to draw it a little Asa. You can understand. I'll just draw it something like this. Okay, now Chlamydomonas, the first step in Chlamydomonas, let me just get rid of this so we have some space for me to draw and write. Okay, now let me draw again. Okay, so here I have Chlamydomonas. Okay, Chlamydomonas, and it has a flagella. Imagine this is the parent cell. So the first step in reproduction in Chlamydomonas, this is which zoospore. The first step or formation of zoospore is that the parent cell loses its flagella. Okay, so the parent cell does not have flagella anymore and becomes non-motile. What does flagella help in? Flagella helps in movement. Okay, motile means movement, right? Movement. So when a Chlamydomonas loses its flagella, so for example, if somebody loses their legs, then they cannot move. They turn non-motile. Same like that, Chlamydomonas loses its flagella and becomes non-motile. They cannot move. Then what happens? This protoplasm. What is the protoplasm? Let's go up here. Protoplasm is basically the entire inside of the cell. So this entire thing here, the entire inside of the cell is a protoplasm. So what happens to this protoplasm? This protoplasm, it divides mitotically. Okay, it divides mitotically into 2 to 16 daughter protoplasts. Means it keeps on dividing. So if I have here the protoplasm, suppose this is the protoplasm, it will keep on dividing. So if I have here the Chlamydomonas, I'll draw it as a circle. Okay, so inside this Chlamydomonas, just imagine it's a Chlamydomonas, I'll just draw a circle. Okay, so inside this Chlamydomonas, what happens to the protoplasm? It divides. Okay, it divides mitotically into 2 to 16 daughter cells. So I have here 2 daughter cells, and I have 16. It depends. Okay, so it divides into 2 to 16 daughter protoplasts. Okay, daughter protoplast. Protoplasm's small bit is called protoplast, each of which develops a flagella and is called a zoospore. So what happens if I take this Chlamydomonas? Okay, if I take this Chlamydomonas and I zoom in a little closer, this is the Chlamydomonas, right? Its daughter cells divides into 2 to 16. Here I'll just draw 2 for understanding. Okay, so imagine this is the protoplast divides into 2 to 16. Now look at this. Imagine this is a Chlamydomonas. This outer layer is the parent cell. Okay, and this inside, these are the daughter protoplasts. What happens to these? These develop a flagella. Okay, these develop a flagella, and what do they become? They become motile, and these daughter protoplasts, which was formed from the protoplasm of the Chlamydomonas, is known as, what is this known as? This is known as zoospore. These are zoospore. So Chlamydomonas, they reproduce from the zoospore. Now after time passes by, oh no, what happened here? After time passes by, what happens is that this mother cell, the parent cell, ruptures. Okay, so if this was the parent cell, a part of the parent cell ruptures, and these zoospores, they are released. Okay, now they don't have a parent covering them. Okay, they don't have anything. They are just released into the water. Okay, and these daughter protoplasts, both of them, these daughter protoplasts, they develop a cell wall. They get a cell wall. Okay, they develop a cell wall. These zoospores, they develop a cell wall, and then what happens? They grow into an adult Chlamydomonas. Each one grows into a separate adult Chlamydomonas. Okay, and in the end, what happens? They grow into adult plant and finished. The reproduction process is over. One important point to note is that after the zoospores are released, or after they get out, the parent cell is no longer there. The parent cell had to burst. The parent cell had to sacrifice itself in order to release the zoospores. So this is zoospore formation or reproduction by zoospore in Chlamydomonas. Now, if we continue, we'll go to aplanospores. Okay, asexual reproduction by aplanospore. The step of asexual reproduction, okay, or the step of hypnospore, the first few steps is the same as aplanospore, but aplanospores only occur if there is a thin film of water. If there is a thin film of water where swimming is not possible, then Chlamydomonas, they are produced by aplanospores. One thing to note is that aplanospores are thin-walled and they are non-flagellate daughter protoplasts. Okay, what are aplanospores? They are thin-walled, non-flagellate daughter protoplasts. Zoospores, they are what? They are thin-walled, but they are flagellate, means they have flagella. Here, they don't have flagella. Aplanospores have no flagella. Aplanospores, they don't have any flagella. Okay, aplanospores, they don't have any flagella and they are thin-walled, same like zoospores, they are thin-walled. Okay, now what happens? The first step is same. What happens? We have a parent cell. Okay, we have a parent cell, a mother cell here. What happens to her? She loses her flagella. Okay, after she loses her flagella, what happens? She turns highly extended. So if this is her, she's a circle, she turns highly extended, she stretches out. Okay, and the the protoplasm inside, which is the content of the parent cell, the protoplasm inside her, would happen, this will divide again mitotically. How many times? This will divide 100 or more times to form more daughter protoplasts. So if this is the mother cell here, okay, this is the mother cell here, she's highly extended. Okay, she turns highly extended, and wait, let me just draw the mother cell. Okay, this is the mother cell here. It lost its flagella, she becomes highly extended, and inside her you get 100 or more daughter protoplasts. Like here, there's daughter protoplasts here, here, all over. Okay, so what happens? And each of these are called aplanospores. Each of these are called aplanospores. Here in zoospores, what happened? They developed the flagella and then they were called zoospores. After developing flagella, they were called zoospores, and they only divided 2 to 16 times. But here they don't develop a flagella, no flagella, and what happens? They are called aplanospores without developing a flagella, and they divide 100 or more times. Okay, so it varies. Aplanospore is different from zoospore. Okay, now what happens? Okay, the parent cell loses flagella, becomes highly extended, and it's called aplanospore. Now, one thing we need to know is that we'll come back to palmella stage. Okay, now leave this. Okay, if this stage is flooded by water, okay, if this stage, okay, is flooded by water, what happens is that the zoospores here, they develop a flagella. Aplanospores here, they develop a flagella. Each of them develop a flagella, and they come out of the parent cell. They come out of the parent cell, they develop a flagella, and they grow into a new independent plant. Okay, now this stage where before they are released outside the water, when they are 100 daughter protoplasts inside the parent cell, this stage it resembles one type of algae. Okay, it resembles a non-motile colonial algae called palmella. So this stage is known as the palmella stage of Chlamydomonas. Okay, so if the palmella stage was flooded with water, what happens? The aplanospores, they develop a flagella and they come out of the parent cell. Okay, so that is how aplanospore forms. Now, if the aplanospores are inside the parent cell, 100 or more daughter protoplasts, if this stage is not flooded by water, okay, if this is not flooded by water, and the water suddenly dries up, what will happen? Some of these aplanospores, some of them will develop a thick black or brown wall. Okay, some of them will develop a thick black or brown wall. Okay, and these which develop thick black and or brown wall are known as hypnospores. So hypnospore is the third asexual method, right? How many asexual methods were there? Zoospores, aplanospores, and hypnospores. So the third asexual method is hypnospores. What happened is that these hypnospores, they develop these aplanospores, if the pulmonary stage is not flooded by water, these aplanospores, what do? They develop a thick wall which are known, and they turn into a hypnospore. Okay, when the favorable condition returns, like when water is available for swimming again, these hypnospores, what will happen? They rupture, they release the protoplast, which is aplanospores, and these aplanospores, they what do? They develop a flagella. These aplanospores develop a flagella, and after these aplanospores develop a flagella, they are called zoospores. And what happens to zoospores when they are released in water? Zoospores ultimately grow into an adult plant. Here also they ultimately grow into a normal Chlamydomonas plant. So this is three ways these are the three ways reproduction is done asexually in Chlamydomonas. Now let's go for sexual reproduction. Sexual reproduction is also done by three different ways. Okay, in Chlamydomonas, sexual reproduction is done by isogamy, anisogamy, and oogamy. And this depends on what? This depends on the species. We learned earlier that asexual reproduction, it depends on the availability of water. Here, this one, it depends on the species. Now let us understand the first type of species. The first type of species is Chlamydomonas eugametos and Chlamydomonas ehrenbergii. Okay, this is the first species, and what do they exhibit? They exhibit isogamy. Now what happens in the first stage? As I told earlier, all the first stages of what? Asexual reproduction starts by losing flagella, right? Every single one, they lost flagella. What? In zoospore, they lost a flagella. The parent cell lost flagella. Here also in asexual aplanospores, also they lost flagella. Aplanospores, the first stage of aplanospores, also the first stage for hypnospore. So obviously hypnospore also lost its flagella. So here, same like that, in reproduction by isogamy, what happens? The parent, the male and the female cell, they become non-motile. How do they become non-motile? Non-motile means no movement. What helps them to move? Flagella. If we lose our leg, how do human beings become non-motile? How can they not move? If they lose their legs, right? So same like that, they will lose their legs, which is their flagella. Then what will happen? The daughter protoplasm, again from here, how many? Here also in asexual reproduction also the daughter protoplasm divides. In zoospore it divided 2 to 16 times. In hypnospore and in aplanospore it divided 100 or more times. Now in isogamy it divides 32 to 64 times. Okay. Each daughter cells develop a flagella and are released by water by the rupture of the mother cell. Now, in asexual reproduction, there is only a single mother cell, right? Asexual means one parent. Here, however, there are two parents. Okay, so I'll draw here the first parent. I'll draw here the second parent. Okay, now this is the flagella. So the first step, what happened to the parents? They both lost their flagella, right? They both lost their flagella. After losing their flagella, what happened to them? Their protoplasm. This is the protoplasm, right? This protoplasm was, what happened? It started to divide. Okay, so let's go over it again. Okay, so this is a parent cell 1, parent cell 2. Okay, the first thing what happened? They lost their flagella. Right? After, how did they lose flagella? They lost their flagella. Now they become non-motile. Now what happened to them? This is their protoplasm. This is their protoplasm. This is also their protoplasm. This protoplasm, it divides. Okay. Protoplasm divides. How many times? Protoplasm divides how many times? 32 to 64 times. Where is it? Here, 32 to 64 times. So the protoplasm divides into 32 to 64 times. So now we have 32 to 64 daughter protoplasts. This is the parent cell, here 1, here parent cell 2, and inside the parent cell you have 32 to 64 protoplasts. Here the inside of the parent cell is divided into 32 to 64 times. Now what happens, this parent cell, what happens is that it ruptures, each of them rupture. Okay, so this parent cell wall ruptures. So here I'll open one parent cell and I'll open the other parent cell. Now what happens to the inside, when the parent cell ruptures, these protoplasm daughter protoplasts, they are released into the water. They come out into the water, and each of them, each of these daughter protoplasts, they act as a gamete. They act as a gamete. Now these daughter protoplasts, what happen, they will be floating around in the water. Okay, when they are floating around in the water, two of them, here suppose this is from parent A, okay, and suppose this is from parent B. Okay, these two from different mother cells, they meet. Okay, what happens to them? From different mother cells they meet. Okay, and they fuse. They develop a flagella. Okay, I forgot to mention here, when they are released here, they develop a flagella. Okay, when they are released, they develop a flagella. Don't forget that. When they are released, they develop a flagella. Okay, here flagella. Now this is parent A and parent B. Now what happens to these two? These two, they fuse. These two become one. Okay, these two become one, and when they become one, what happens to their flagella? They have 4 flagella, here 2, here 2. So they become quadriflagellate. Quadriflagellate what? Gametes. Okay, now quadriflagellate zygote or gametes. What is that? Fusion of two male and female gamete. What happens? They turn into a zygote. Okay, now these two contents, when they fuse, okay, now this is haploid. Chlamydomonas, what is Chlamydomonas? Chlamydomonas is all haploid. Say this is 2N, so that's diploid, sorry. This is N, this is N, right? Haploid. These are also N, these are also N, which is haploid. Haploid means 23 chromosomes. This is also N, this is also N. Inside each of these daughter cells are also haploid. Each of these daughter cells are also haploid. And this is also N, this is also N. But when they fuse, when they're content, when they're inside, so if I take 23 chromosome plus 23 chromosome, which is haploid plus haploid, what happens? They turn into diploid. Means they have total 46 chromosomes, right? So this quadriflagellate has 46 chromosomes, and this stage, this is the only stage in Chlamydomonas' life cycle. Which stage? Quadriflagellate zygote stage is the only stage of Chlamydomonas' life in which it is diploid. Means it has 46 chromosomes. Only this stage. Now what happens after this stage is over? What happens when the content fuses? After the content fuses, the zygote develops a thick wall around itself, which turns brown to black. If the condition is unfavorable, what happens with this? They form a, like how hypnospore had a thick wall around them, same like that, they form a thick wall around them. Now when the condition returns, what happens when the condition returns? On the return of favorable condition, which is temperature, food and water, the diploid nucleus of the zygote divides by meiosis and forms 4 haploid zygote. Now this is diploid, right? So what happens? They split. Okay, they split and they form 4 haploid zoospores. The nucleus, which is diploid, splits, and this is the nucleus. Okay, it is diploid. It splits into 4. What is this? These are zoospores. And how do zoospores grow? We learned that zoospores grow into a normal Chlamydomonas adult plant. Normal Chlamydomonas adult plant. So this is how isogamy is done in Chlamydomonas. Okay, these are the steps of isogamy in Chlamydomonas. Now we can continue with anisogamy. Now anisogamy, again, isogamy was done in what? Chlamydomonas eugametous and Chlamydomonas ehrenberg. Here anisogamy is done by Chlamydomonas browni. Again, from the beginning, what did I say? The male and female lose their flagella and become non-motile. Don't need to go through much into this. This time what happens is that the male protoplasm, it divides into 32 to 64 biflagellate gametes. So it divides into 32 to 64 biflagellate gametes, whereas the female cell produces 18 to 16 biflagellate gametes. Means the male cell is small and the female cell is large. Okay, both these male and female, again, they are ruptured and they are released into water. Same way how this was formed, same way they are released into the water, and the larger females gamete lose their flagella and they become non-motile. Biflagellate means two flagella. Okay, so this is two flagella here also is two flagella. So in male protoplasm divides into 32 to 64 biflagellate gametes. So they form 32 to 64 biflagellate gamete. Here only 16 to 8 to 16 biflagellate gametes are formed. Same way the parent cell ruptures as it was done in isogamy. The same way the parent cell ruptured, the male and the female gametes are released in water. Then what happens? The large female gametes, they lose their flagella. The female gametes, which is large, they lose their flagella. Okay, and they become non-motile, and each is fertilized by a male gamete. Okay, after fertilization the fusion product loses their flagella. Now after fertilization, how many flagella will they have? Now the female, she doesn't have a flagella. The male is smaller and they have a flagella. So if they fuse, then we have two flagella. Now this one, it loses its flagella and it becomes spherical. It turns into a circle and develops a thick wall. Okay, same like hypnospores, this develops a thick wall during unfavorable condition. When favorable condition returns, what will happen? The zygote undergoes meiosis, it undergoes division. Then it forms again 4 haploid zoospores here. It forms 4 haploid zoospores, and these grow into normal Chlamydomonas plant. This is very easy, very simple. No need to go too much in detail. Just the same method repeated again and again and again and again. Nothing much to remember. Okay. Now let us go for the final zoospore development, that is final sexual reproduction by oogamy. Okay, now oogamy is exhibited by what? It's a sexual reproduction exhibited by Chlamydomonas cosifera and Chlamydomonas visi means Chlamydomonas ooganum. Okay, here the male and female, again, what did I say? The male and female cell loses their flagella and become non-motile. So Chlamydomonas, first step in reproduction of Chlamydomonas is it always loses its flagella. Okay, first step is always losing its flagella. Okay, then what happens after it loses its flagella? What happens here? All the content of the female cell act as the female gamete or egg, same like it was acting. The same thing was happening here in isogamy, the same way, in anisogamy also the same way, in oogamy also the same way. Okay, but the protoplasm of male cell divides into 32 to 64 biflagellate gamete. So the female cell, she remains as she is, and her whole content acts as what? It acts like a gamete. She remains as it is and her whole content remains as a gamete. But the male, again, they divide into 34, same like they divided for here, they divide here again to 32 to 64 biflagellate gamete. These biflagellate gametes are liberated in water, means they are released in water, and they swim around in search for female gamete. Okay, they swim around, same like they swim around all the time, they swim around here, and when they find a female cell, 2 or more enter the female cell and having a non-motile egg, but only one fertilizes the egg, the others just degenerate and they contribute to the nutrition of the zygote. So if we have here a female cell, she turned non-motile and she is here. The male cell, they have small little spores, biflagellate gametes going around in the water. These three, they will find the female cell and they will find the egg and they will go inside and they will fertilize. Only one of them will fertilize, the others, they will die or they will degenerate and they will feed the developing zygote. So which one is fused? These will die and they will act as a nutrition. The fusion product of the egg and the male and the female of egg and the motile gamete is called zygote, that we know, that develops a thick pigmented wall to enter resting phase. Everything has a resting phase over here. Okay, when it's unfavorable condition, what happened to the resting phase? They have a black wall surrounding them. Okay, same like hypnospore. Okay, on return of favorable water and condition, these zygote undergo meiosis and they produce 4 haploid biflagellate zoospore. They form zoospore, each of which grows into a normal independent plant. So what happened here in oogamy, they had 4 haploid zoospores. Here also in anisogamy also we had 4 haploid zoospores, and finally in isogamy also we had 4 haploid zoospores. So in isogamy, anisogamy and oogamy, we can say that 4 haploid zoospores is the ultimate formation, like this ultimate goal, and then from that we get our Chlamydomonas. So this concludes our topic for today. If you look at the diagram carefully here, you can see you can see the different type of zoospore formation, and you can understand by reading these, you can understand how and which you can visualize it in your mind, which is isogamy, which is anisogamy, which is sexual reproduction and asexual reproduction. In the next class we can go ahead. See, this is anisogamy, this is anisogamy, and this is oogamy, and this is, that's it. Okay, so next class we are going to do Spirogyra. We're going to be discussing its structure and its reproduction, just like we did in this class for Chlamydomonas. That concludes our topic for Chlamydomonas.